Hi, today we want to focus on a special lab, an activity we will do in class. It looks like this. Can you tell what's inside the bottle? Can you see it? You will be working today in this lab with crickets. And you may notice they have a little bit of color on them. You're going to actually use paint pens to mark a cricket, a live organism. You will have a few that you will collect and paint the top of their exoskeleton, their thorax region, or their abdominal area. So this lab is called Mark Recapture with crickets. But before we can do this, let's learn a little bit about the lab and why we're working with crickets. It's fun and all, but we need to know why. We're studying crickets because we want to understand this scientific problem. It's how can we know when our ecosystems have an environmental problem? How do we know if there's a problem in the environment? Well, one of the main ways we know there's a problem is when we see a population of a species go down. This species, for example, there's only 670 giraffes, this species of giraffe, 670 for a population. This species is threatened. The environment or the ecosystem this giraffe lives in, something's wrong. There's a problem because this species is going extinct. It's close. It's being threatened because there's very low population. Same for all these species. There's not a lot of these species. These are all endangered species. So how does biology investigate this problem? How does biology find out how many there are of these species? How do we know there's 670? Well, we're going to practice an important method called Mark Recapture. And you're going to do it with crickets. This lab is all about studying threatened populations. So threatened populations, different species like elephants, gorillas, pandas, and giraffes. So we're studying threatened populations. We want to measure threatened populations. These are all reasons, density dependent and density independent, are all different reasons why populations are threatened. Climate change causes populations to go down. And parasites, competition, lack of food causes populations to go down and become threatened or endangered. So let's think about a special organism, the bee. The bee is a current organism on Earth, and there might be a problem with it. They might be threatened. Let's check it out. So here is a man who grows bees. His business is to have bees, but his beehives are almost empty. He's finding very few bees in his farm, and he's stressed. He's like, I don't know where the bees are. They're dead or missing. So he can't find most of his bees. And he uses bees to make money. He uses them to make honey, and he uses them to pollinate flowers and crops. So his bees are his business, and they help him make money. It says right here, empty hives. Look at, these are all boxes of bees empty. The bees are dying or they've disappeared. These are researchers investigating to understand what's happening to the population of bees. Why is the bee population crashing? And these guys, they work with bees. That's their business, and they're confused. So here's another video, and the title of it is called The Death of Bees Explained. So we're finding that the population of bees is declining. But bees play an important role. When bees are dead, plants die too. So this is why bees are so important, and this is why humans care about the population of bees. Because when bees die, plants or crops might also die. Bees right now, all the bees we use, they're responsible for the world producing $173 billion. So if it weren't for bees, we would have $173 billion less. And it's going up. Look at this. It's gone up all the way to 265 billion. And why is that? Because we need bees to make fruits and vegetables everything we eat. There's meat here too because cows eat what we grow. So food, we don't have our food. We don't have fruit, we don't have vegetables or cattle without the bee. So they're quite important. So if bee populations are crashing, humans care too because they're worth so much money in making honey and helping us grow vegetables to eat, to feed people. And if we have a lot of the world starving, you can see the non-blue colors are people that are going hungry. They need food. They need fruits. They need vegetables to live. And we have a lot of places where people are starving. Well, if bee populations are crashing, human populations might also crash. So we need the bees so we can make the vegetables and fruits to feed them. So we're going to learn a special technique that allows us to measure a population by marking them. You see the little mark right here? Here's a paint pen marking. This bee has been marked. And we use this technique called mark recapture to measure the population of threatened species. So we are going to mark the crickets. So the mark recapture method, I will purchase and provide a population of crickets. Your team are going to get 2 crickets or more, depends, but you're going to get some crickets to mark with a paint pen, this thing. So you're going to have a collection of crickets, a paint pen, and you're going to mark them. And then you will enter special data into the Google Classroom spreadsheet called Studying Threatened Populations. The first thing we'll do, our first day of mark recapture, is trapping and capturing them. Every individual you're going to mark. So all the crickets you will mark. So this is unmarked. You're going to mark them though with a paint pen and you'll color them. So they have a new color. So you're going to take your paint pen and mark the organism. So pretend these are the crickets. You're going to mark them. Just like this cricket. This cricket was marked with a yellow paint pen. So paint has marked this cricket. So this cricket was captured and it was marked. Next, after you mark them all, we release them back into nature. So we let them out of the cage and we let them go. So there's going to be a bunch of crickets let go with a bunch of markings on them. On our second day, we will recapture them. We will capture a bunch of crickets. Each team will have a bunch of crickets, and some will be marked, and some will not be marked. Your job the second time is you're going to count the number marked. 1, 2, 3, 4. 4 marked. And you're going to enter this data into the spreadsheet. So just like this is data from last year, and you're going to enter how many crickets you marked. You can see each team here marked 2 crickets. So day 1, this is the number of animals the team marked from their first capture, and everybody marked 2 crickets. So they painted 2 crickets. Day 2, you're going to count the number of animals with no mark, and the same day, our second day, you're going to count the number of animals captured that have a mark. And you'll put in how many you caught that have no marks, and how many you caught that have some. On that same day, you will then estimate the population of crickets just based on that math. You will predict the size of the population with only that information. And that will be N. N equals the estimated population size. So using the spreadsheet, you will calculate N, or you will estimate the population of crickets using the spreadsheet. So you'll go to Google Classroom, open up the spreadsheet Studying Threatened Populations in Google Classroom, and then you'll enter in that data and calculate it. And eventually, you'll have an estimation. Last year, period 1 predicted there was 283 crickets. Later, you will decide how far off you were from the correct number if you had percent error. But to be clear, on day 1, you are going to mark some animals, and however many you mark represent N1. this number. So if you marked 2 animals, you would put a 2 here. On day 2, you are going to count how many animals have no marks. In 2, you would put that number right here. Next, day 2, you need to find this number, M2. M2 is number of animals that you captured that had a mark. That goes in the denominator. If you take N1 times N2 and then divide it by M2, it equals the population size of the crickets. It will tell you how many crickets are in the total population. It will estimate all the crickets in the container. And there's a lot of them. There's hundreds and hundreds and hundreds of crickets. But just with this simple math, you'll be able to estimate the population size of the crickets. Later in class, after we do all the data, and after we calculate what we think the estimated population size of the crickets are, using this little equation, we will then analyze the results. We're going to analyze this threatened population of crickets. You'll put your name, you'll put everyone on your team, you'll choose your class period, and then you'll go to your spreadsheet. It says open and use Google Sheet that we used. It's this one, so you'll open this up and look at it and use it to answer questions below. It says calculate the percent error using whole class data. We'll talk about this later. You will compare and contrast your class period percent error to the percent error of the whole school, all periods together. Finally, you will say which sample, class period data, your classes, or the whole school's data is more reliable, which had a lower percent error, and you need to use evidence in your argument. So you'll have to use information from the data sheet. So you'll have to look at your percent error for your class period compared to the percent error of the whole school data, and you will have to identify which was better, the whole school or our class period, and you have to use evidence from the spreadsheet in your argument. So which had lower percent error. And then finally, you simply have to put how can mark recapture methods be used to study endangered animal populations, like these animals here. How can we use mark recapture to study endangered species that are threatened and have very low populations? Here's another example. We mark some organisms like these turtles. We mark them just like you see here, and we can then identify the number of animals in the population, and we can know if the species is threatened or not. Because if their population is going down, we know that they're threatened. And that is how we answer this problem. Mark recapture, doing mark recapture, helps us know when an ecosystem has an environmental problem. All right, thanks folks. I hope you've enjoyed this, and I hope you're ready to do our lab. I hope you're looking forward to it. Have a good day.